My name is Ivânia and I'm currently a PhD student of IMTech, the Institute of Microsystems Engineering at the University of Freiburg, and I'm also located at ATLAS Neuroengineering. My PhD main focus is the design and development of a flexible microelectrode array that can be used for long-term recording and stimulation, and also the design and development of insertion techniques that allow the implantation of these electrodes in the deep brain regions, namely the brainstem of the macaque brain. My institution itself does not have animals in its facilities. However, we do validate our neural probes through collaborations with universities that have animal facilities and perform electrophysiology. Microelectrodes are electrodes whose dimensions are in the order of micrometers. If we think about the analogy of an elephant communicating with an ant, the elephant will have a hard time listening to an ant speaking. A conversation between two ants would be much more understandable for both of them, because they are the same size. The same happens with neurons and electrodes. If we have electrodes that approximately match the size of the neuron, we will have a much better conversation between both of them. So we'll have better spatial resolution and also single-cell recordings. With regards to motor control, the brainstem is like a relay station for motor commands originated in the motor cortex that is located at the brain surface. These commands are retransmitted to produce coordinated movements. So by implanting electrodes in the brainstem, we can directly record and study the underlying mechanisms involved in this process, and this can give us some insights into certain diseases, such as the motor neuron disease. The macaque brain, brainstem included, shares many similarities with the human brain in terms of anatomy, physiology, and neural organization. Therefore, studying the brainstem in animal models, such as the macaque monkey, provides insights that can be translated to humans and will pave the way to translate research results towards clinics and hospitals. Well, for me, the moral hurdle lies within the ethical compromise, because balancing the potential benefits of the research outcomes with the welfare of animals is a complex dilemma. We need to advance science and technology to make life better, to understand diseases and to find cures and treatments, and we need to do this without causing any suffering to the animal. So I think it's very important to communicate in a clear and direct way with all of the parties, such as the neuroscientists, the veterinarians, the ethical boards, as well the general public, to foster not only transparency, but also to foster trust in the researchers, and make people more familiar and aware about the importance of using animals in research.